Well, welcome back to the lab, guys. Today we're going to be talking about the role of buffers. You might want to ask, what is a buffer? And that's a really good question because that's what we're going to be studying today. What are buffers? How do they work within mammalian body systems? And in particular, what makes up a good buffering solution? Now, one of the things you're going to have to do is come up with a hypothesis, and we're going to be comparing two different kinds of solutions. We're going to take a look at just plain water, and then we're also going to be taking a look at club soda. This particular brand has a variety of different solutions in it, and you are going to be testing these solutions plus water, and which one makes a better buffering solution. Well, all creatures, even if they're single-cellular or multicellular animals, need to maintain a very stable conditions in order to survive. For instance, they may need to maintain those healthy levels in their temperature, their pH, and also their water content. So those are the three things that have to kind of line up to be able to maintain this healthy system. The other thing is stable levels in metabolic conditions. It's called homeostasis. Homeostasis is that kind of that place, that level on which mammals and just all cells can maintain this healthy living and healthy level. For instance, most multicellular organisms really contain a very complex order of buffering systems. And this buffering solutions, what they try to do is they resist change in pH levels. They have certain kinds of solutions that prevent it from going out of whack. So we're going to be learning about that today. And for instance, mammalian body, the pH is maintained by a variety of different substances, primarily the carbonic acids and bicarbonate solutions. So those are the things that help level things out and maintain that homeostasis. So in order for us to take a look at this, we need a variety of different equipment in our lab, and I want to show you that before we get started on our lab. One of the things you're going to need is you're going to need some water. So we just get standard distilled water, and you'll need a 250 or a 100 ml beaker, whichever one you have. I like the 200 ml or 250 ml just because it's a little bit smaller. So you're going to need also, you're going to need 100 mLs of water. So when you're reading through all of the directions in this lab as you get your data loggers, you're going to be measuring out 100 mLs of water. Now I don't want you to take this beaker to the sink and just fill it up. What I want you to do is I want you to use the graduated cylinder. That way, by using the meniscus, we can get an exact amount. This isn't very accurate. This is accurate. So we're going to go ahead and just pour that 100 mLs of water right into there. So we're going to use a 200 ml beaker, get 100 mLs of water. We're also going to use this stir plate. We have a variety of different kinds, but this is our newer variety that we have. Obviously, make sure it's plugged in, and then you have the ring stand built into it. So you're going to put this right on the stir plate, and then make sure you get a magnet, because a magnet is going to spin on the inside of the beaker because of, let me see if I can get it to move. I don't know if you can see that, but that's going to spin just like that inside our beaker and basically mix the solutions as we put them in. So I'm going to put that in there, put that right in the center, and I'm just going to turn it on and mesmerize you as we go through this. Okay, now once you've got that done, so we need a ring stand, we need a beaker, we need 100 mLs of water. You're also going to need some vinegar, so we're going to be using this again, our graduated cylinder, and I just poured a little bit of vinegar right out of the 1 gallon container right here. This is the vinegar. And I'm just going to measure out 10 cc's or 10 mLs. All right, so now what I've done is I've gotten my 10 mLs of vinegar, my 100 mLs of water, in a 200 to 400 ml beaker. I have it on a stir plate with a magnet on the bottom. But what are we going to measure? Well, remember we talked about buffers really resisting changes in pH, because pH is really important as far as the cells taking in or not taking in certain kinds of elements or nutrition. So we're going to be needing to measure the pH. So we're going to be using this Pasco pH probe, a digital probe, and the magnifying box. Make sure it says pH here. These two connect by a coax cable, or at least it looks like a coax cable. And then, of course, you'll need a data logger. The handheld variety works, or the one that we have, a SparkLink. And this SparkLink is connected to a lot of our computers in the classroom. So you need to decide whether you're going to do SparkLink or the data logger. This one's much more flexible on where you're going to be sitting in the lab, whereas this one's kind of tied to the computer, but this does have this advantage is it's a little faster, you can save it easier, and of course you can print right from your computer. This one saves as well as prints, but it's not quite as easy to make that happen than it is on this. But you decide, it's up to you. I'm going to go ahead and use the handheld data logger. We'll take the handheld data logger, take the amplifier box, connect it inside the port, and now we're ready. Now what I'm going to do is I'm going to place this right in here. So I got the pH probe right on the ring stand. This should be lowered into the water so that the bulb on the inside is completely covered in water. Now what I'm going to do next is I'm going to go ahead and hit record. And then what it's going to do is it's going to record the pH of this water for a good amount of time, 30 seconds to be exact. And once that's done, then what I'm going to do, I'm going to continue to let it run. Don't turn it off. Let it keep running, and then I'm going to pour in all of my vinegar and let that run for 10 to 15 seconds. Then stop it. So it's really important that you follow directions. I'm going to let you catch up to me, get all your stuff together. Then I'm going to run this and see how you do. All right? So make sure you get your data logger, your pH amplifier, the pH probe. You'll need this 200 ml or 400 ml beaker, 100 mLs of water, your stir plate, 10 cc's or 10 mLs of beaker, or excuse me, of vinegar, and you should have everything ready. So that's the first part. The second part, we're going to use club soda. So make sure you get all the parts together, and we'll see you in just a minute. Thanks. Well, we're back again. This is the second part of our lab. We're going to be taking a look at club soda and vinegar. On the first part, we looked at water and vinegar. And before we go to the second part, make sure you make the prediction in your data logger. Which one do you think is going to be a better buffer solution? Water by itself, or is it going to be club soda and vinegar? Which one is going to be a better resistance to changing pH? So make sure you do that now before you can begin the second part. Now the second part, you're going to start with that ubiquitous 200 ml or 400 ml beaker. And this, make sure, by the way, when you are finished with beaker number one, you rinse it all out. And when you rinse it all out with the distilled water, we'll have a lot less residue left over. So anyway, you're going to take your beaker. Going to make sure you have your magnet in it, and then what we're going to do is set that aside for just a second. So we have our 200 ml beaker or 400 ml beaker. You have your magnet, you have your stir plate. You're going to also get some club soda, and you're going to need 100 ml. So let's go ahead and get that. I'm going to push this out of the way for just a second, and I'm going to get a graduated cylinder. Now I've poured a little bit of the club soda in this smaller beaker, and I'm going to pour out 100. And when the meniscus is on the bottom meniscus on the top line, the 100, I've got that. So I've got 100 mLs of my club soda. I'm going to pour this right in the 200 to 400 ml beaker. In this case, this happens to be 400. Got that all done. I put it on my stir plate. I've got my stir plate with the ring stand built in here. Now the other part of this is I'm going to use of course, my data logger and my pH probe. I'm going to put the pH probe right down so that the bulb is covered. I'm going to go ahead and, shouldn't turn it on too high. You just should be able to stir it just a little bit. If you're creating too much of a vortex, like I'm doing right now, it's not going to it's not going to allow me to really cover the bulb very well. Okay, there we go. So, again, you have your stir plate, you have your pH probe, you have your magnet, you're going to have 100 mLs of club soda, and you're going to have 10 cc's or 10 mLs of vinegar. You're going to let this run for 30 seconds, keep it running, don't turn it off, and then you're going to pour this after that 30 seconds. Then wait 10 seconds, get the reading and see how much it changes, and then hit stop. So follow the directions as they are in the data logger. You should be able to come up with some pretty good data at the end of this. Now, please remember, you're comparing water's ability to act as a buffer versus club soda. Remember, the thing that we're adding that's constant is the vinegar. Vinegar has a certain kind of pH, and I'm not going to tell you what it is, and we're going to see which one of these substances, club soda or water, has the best buffering capacity of the two. So please make sure you snapshot or save all of your graphs as you do them, because that's going to be really important in the analysis. All right? So we'll be getting back to you on the other side of this lab. Thanks. Bye. Well, what'd you find out? Is it water the best buffer solution, or is it club soda? Whichever one it is, you have to take a look at the substances that are in each of these and determine what is it that's in it that makes it a good buffering solution. Now remember, when I first started with the background material, I said that a good buffering solution is one that helps resist change. So which one of these had the least amount of difference between the two, from initial to final? That's what your job is when you're analyzing all of the graphs. And before you turn in your lab, please make sure you understand that the one that's going to have the most resistance to change, is it going to be a greater difference or a smaller difference? That's what you got to determine. All right, thanks.